My name's Viive Howell. I'm a molecular geneticist and a cancer researcher across the road in the Kolling. And I'm standing here in front of a slide talking about an irradiator. So why is that? When I'm a molecular geneticist and have got nothing to do with, have no background in radiation. So I work in Neuro-Oncology, so we work on brain tumours but also on brain metastases. And when we're looking at developing new treatments, what we really first need to do is put those on a background of current treatment. And what you've heard tonight is that, as you can see, a lot of that is radiotherapy. And for the researchers at the Kolling and in a lot of places, we just have to, up until now, we've just had to totally ignore the fact that that radiotherapy exists when we're trying to develop new treatments, because we have no way in the laboratory to recapitulate radiotherapy in our animal models. We can certainly do chemotherapy, we can do targeted therapy relatively easily, we can do surgery, but we just have not been able to recapitulate radiotherapy because we have no irradiator. So that's why I'm standing in front of you with showing you this particular slide. So last year, a long time ago now, we put in a grant, a group of us put in a grant to get an irradiator. And we weren't able to do this without the support of the radiation oncology department, because we needed to have some co-funding for this. But it was really about time that, that we got an irradiator. And so we put in a grant to Cancer Institute, and it was awarded to us. It's to date the second largest equipment grant that the Cancer Institute has ever given out. So it was well over 800,000. And we needed to have co-funding, as I said. A lot of things changed this year in finance across the road. So we have co-funding from Northern Sydney Local Health District and from the University of Sydney. Very ominously, the equipment arrives on 9/11, so I'm not sure what that means, but that's when it arrives at our lab on the 11th of September. There's commissioning and installation that week, and we'll start doing training and have a launch after that. So it will be housed in our animal facility, and it will be the first of its kind in Australia. There is one other image-guided irradiator, preclinical image-guided irradiator, but that's a different brand, and that's over in Perth, which is a bit far for us to take our animals. We can't even take our animals out of the animal facility to University of Sydney or across to the hospital and then bring them back again. It's just one of those things about working with animals. So when we were looking at getting an irradiator, I talked to the radiation oncologist, Michael, and Tom Eden. I'd been pestering Tom for a while saying, you know, how do we do this? What do we do? How can we make an irradiator? And he said that, well, there's this new one around. And I talked to Michael and he said, well, you know, a whole body one is just not where it's at these days. You really need to have a focal image-guided irradiator. So I'll just, this is what it will actually do. But why would this be of interest to you? Well, I'm hoping that you have questions that you need to ask that you can't actually ask in your patients. And Alex has already mentioned some of these, but just off the top of my head, you know, sequencing of radiation with immunotherapy, with surgery, what's the right order? Should you be doing surgery first? Should you be giving immunotherapy first, for example? These are things that we can start to answer in the animal models now that we will soon have this instrument. So sequencing, understanding synergies, looking at the complications, looking at the toxicities. We can also then start to look for biomarkers of response. And while we're working with animal models which are somewhat clonal, they don't all react the same way. They don't all respond the same way. So we can take blood longitudinally. You can keep them going when you know that they're not working. Things that are unethical to do in humans, we can be doing those things. Oh, there's nothing. We can be doing those. We could be doing those things in the animal models. So we need your questions. These are some of the things. I mean, abscopal, does it actually exist? Has anyone ever seen it? No? Yes? You see it all the time? So is this, could we hit a met somewhere, could we have an animal model where we've got a tumour in the lung and a tumour, somewhere else, subcutaneously? Can we hit the met? And what happens to the main tumour? These are things that we can start to answer with this model. And the data to date using these pieces of equipment, which are available in Europe and in America and Canada, just not in Australia, is because it's focal irradiation. You can target it very finely to the tumour, sparing normal tissue, which recapitulates much better than anything else what is being done in humans. The results to date really match very well with the human clinical trials. So what actually are we getting? It's called a SARRP, a Small Animal Research Radiation Platform, but an instrument by itself is not enough. It's a very sophisticated piece of equipment in terms of animal studies, but it's not enough. We really need to have experts helping people use these pieces of equipment. And so we have in place already one of the managers. So Kelly McKelvey, who'll take over from me after this slide, she has expertise in animal modelling. And really, if you don't have the right animal model, nothing's going to work. When you're looking at immunotherapies, we can't be using xenografts because they don't have an immune system. You need to be using immune competent models. Now, we have a number of those in our lab. We need to be developing more. We need to be developing better ones. But we have some to get started with. And not everyone can do animal modelling. Not everyone wants to do animal modelling. But this is something that we can give you assistance with your questions. First of all, picking the right model, and also the biology of it, what to look at it. So Kelly is funded by Sydney Vital. She's the Inflammation Flagship Fellow, and she's also partly funded by the Sydney Neuro-Oncology group. So she's working on inflammation and radiation herself, and specifically in brain cancer to start with. So she can help you. She will be the manager of the system, and she has great expertise in animal modelling. But that's not enough. Neither of us have any experience with radiation. So for that side of it, the radiation oncology department are looking at also having a medical physicist to help with the treatment planning to make sure that the system is actually doing what it's supposed to be doing. We don't know that much about it yet. I can't show you any results because we don't have the system. But hopefully, in order to make sure that what you're doing is clinically relevant, the doses that you're giving, the level of the doses, whether it's fractionated or not, that's something that the medical physicists and the Department of Radiation Oncology will be able to help to make sure that you're actually doing clinically relevant work. So I'm going to hand over to Kelly now, and she's going to tell you a little bit more about the instrument.
All right, good evening. as you know, I've joined the Sydney Vital group only about 2 months ago, and in that time, really, I've worked in the background. So it's one thing to get the machine in, but we also have to have the licensing in place and animal ethics in place and some of these other things in order to make sure that once the machine's on site and once it's hooked up, we can actually get on, start looking at what its capabilities are, and also what some of its limitations are so that we can start answering some of those questions. So you see here, we've listed a few of the sort of basic outlines of what we can do with the machine. It has a CT function. This is a cone beam CT imaging. We also have something called bioluminescence. So Jonathan spoke earlier about the sort of tumour paint type idea. What we can do is genetically modify the cancer cells when we put them into the animal so we can specifically see the cells and we can target them directly and very accurately with the radiation. As far as the radiation therapy goes, as Michael's brought up a lot of the time, we actually use fractionated radiation instead of using just a one whole single dose, and we also target very sort of volumetrically and with different doses, so we can do a sort of intensity modulated one as well as the volumetric. We can look at both cell lines, so we can irradiate cell lines to look at more mechanistic interactions, as well as doing whole body. We can look at whole regions, as well as doing the very small focal irradiation. And that's down to a 0.5 millimetre type diameter. Finally, for patients, what we can mimic with this machine is some of the treatment planning, some of the radiation delivery, the doses, as well as trying to protect some of the normal tissue. So with this machine, hopefully we can bring it really into that sort of area of what is being done for the human patients at the moment. So this is a quick video. It's put out by Extral, the company itself. The machine's a cabinet X-ray machine. It's mobile, other than weighing 2.5 tonnes. I won't be lifting it. As you can see, it has CT imaging. This is within the machine itself, so we do this 5 minutes before we do the radiation therapy. I think patients currently have it about 24 hours before. We also have the bioluminescent imaging here. You can see the radiation, the bioluminescent cancer cells will fluoresce there. We can overlay that on top of CT imaging. We can also fuse to that MRI and PET scanning images once we get a machine to do that aspect as well. Focal radiation, you have complete access right around the animal. The animal's anaesthetised the entire time. We can do static. We can also do an arc therapy where it's treated constantly. This reduces the skin exposure to the radiation dose while maintaining a very high dose to the tumour cells themselves. And this sort of replicates the sort of conformal. So not only is the platform moving in the XYZ plane, the gantry is also moving in a 360-degree plane. So we can really get very close to what you're doing to the patients in the clinic. We don't have this accessory at the moment, but this is a variable collimator. It works with an XY frame, so we don't have the multi-leaf, which is very fancy, which you have in the radiation oncology. They are looking at developing one, so maybe in a year or two. Hopefully they'll come out as another accessory for us. So this just gives you a brief example of what the arm to the machine looks like. So the X-ray tube at the top here, we have a maximum of 225 kilovolts, so it's not the mega voltage machine you use for brain therapy. But this fits the Extral medical 300 machine that you have over in the radiation oncology that you're using for skin cancer and lymphoma, I'm told at the moment. So through here we have a couple of filtration units, but realistically the X-ray beam is then filtered and narrowed all the way down through a series of collimators. They're made of brass here, whereas you have a sort of a more cone type system, which I've seen over in radiation oncology department. So you see that the animal's anaesthetised, just lies on a little bed here, and this directs it, and that sits about 6 centimetres from the animal, which means it's very, very accurate, and the sort of isometric accuracy is, I think, about 0.2 millimetres, so very, very small, very accurate, and we can get down to the sort of grey, whatever we're using in the patient clinics, the sort of 20 grey, 60 grey, and we can do that, as we said, in the sort of fractionated patient regime as well. So these are just some schematics that give you an idea of what we can do. We can give a single sort of dose in one direction. We can give separate doses so that we can build up and try and reduce some of that exposure to the skin and to other sort of critical organs in the way, perhaps lung and other sort of colorectal type organs. Obviously not so relevant in the head, but we might be trying to avoid things like the eyes, for example, or perhaps some of those more sensitive, eloquent areas. Again, the arc therapy and that conformal, which looked really cool, where it spun and rotated at the same time. And this is an example here of what can be done. So this is a mouse head. You can see the brain tumour here. And this is the sort of isodose patterning that you see. And you can see that that same sort of pattern that Michael showed you earlier, where we have a very high dose concentrated to the tumour, and we have a very rapid drop-off. So that sort of drop-off or penumbral type area of normal tissue that's receiving maybe 20% of that radiation dose is very, very minimal. And with our collimators, we can obviously enlarge and do a whole brain sort of action as well. Ideally, where we're going is to obviously use this preclinical machine and to really look at the tumour microenvironment. So you've raised a number of points. Obviously, with neurosurgery, we draw up a number of sort of wound healing factors and coagulation type factors. What's the impact there on the brain tumour? And what are some of the other things with the immunotherapies, the anti-PD-1, CTLA-4? That's fine, but what does that do? How does that change the environment? And what does the brain cancer cells, what do they do? What do they react, and how do they react to that? With radiation therapy, again, looking at things, what is some of these long-term effects? Can we look at radiation necrosis, and why does that happen? Which are the cell types? Is there a cell type that sort of dies and that starts things and sort of gets the ball rolling in terms of cytokines and things? And so we can look at that, and obviously, like Viive said, we can look at the sort of sequencing of different drugs, all the way from neurosurgery, radiotherapy, and immunotherapies. And we've brought the neurosurgeons over into the laboratory and given them a mouse to play with. Looking at you, Michael. So here's just some other sort of projects that you might be interested in looking at. Obviously, Viive mentioned a number of the sort of more biological type aspects. That's cool, I'm a biologist, that's interesting for me, maybe not interesting for you. So if you work in sort of radiotherapy or maybe the nuclear physicists or medical physicist areas, you might be interested in some of these other actions, the sort of deformation, so lung cancer here. As you inhale, obviously the lung cancer expands with the lungs, and then when you exhale, obviously it moves. Well, what does that happen? What do we do then with radiotherapy? Do we have to cut the radiation beam on and off, or do we look at just irradiating a slightly larger area? How can we look at some of the other actions? Again, the lung tissue is a very low density tissue, so do we get more radiation scattering than we get in other tissues? And also, what about some of these radio enhancers? Can we try and increase the amount of damage to the tumour cells while trying to minimise a lot of that damage to the normal tissue cells around? And I know that there's areas, a number of work going on in the area there, using some of either the sort of metals or some of the other small molecules and the radiation sort of immunotherapy combined type actions as well. So finally, Viive's touched on this very briefly about where we're up to. As she mentioned, in the week of September, we will be having the machine arrive. It's currently on a ship, and so that'll be trucked over and taken off with a forklift, and then installed into the laboratory. They'll then check all of the sort of dose commissioning, the same as you would if you had a new Linux machine come into the patient clinics as well. An information session, so that's been just arranged at the moment, but that'll be on the 20th of September between 4 and 6, and that actually includes an informative session from one of the extrail application specialists who has expertise working with the machine. So if you've got some questions around what the machine can do, and you don't want to ask me, that's fine, I don't mind. But you can keep all of those really advanced and intrinsic type questions for that application specialist. And we've also arranged that during the training session week, which is from the 18th to the 22nd, that there'll be some time available for our medical physicists and our radiation oncologists to meet specifically with the application specialist to ask some of those questions around the Q&A and the QC type aspects that we need to ensure that our machine's doing what we think it's doing, and to make sure that it's replicating the human setting as well as the standardization of all of those radiation dosages. So here are my details. If you have any general questions about the irradiator, what it can do, if you've got more specialised questions, again, just feel free. You can email them to me. I'll put them directly to the application specialist and see whether they can come up with some answers and maybe incorporate that into their information session that they'll have. But that's all from me, so hopefully you've enjoyed your evening.
Thanks, Kelly. Any questions for Kelly about this amazing opportunity? Viive, do you want to say something?
No, just, we're really interested to know what you think the questions are that we should be answering with this whiz-bang piece of equipment and with our animal models. So we can make up we have our own research questions, but we're really interested to know what you think are the important issues we should be investigating. Yep, Dale.
Yeah, mice, yeah.
Something like this, the question of how we fund it comes up. And like if I had an interesting question, where would I go for funding for this sort of device? Is it ARC? Is it NHMRC? Cancer Institute, New South Wales?
Sydney Vital.
Where do you think we'd go?
Sydney Vital.
Sydney Vital must have that much money.
How much? Oh, a clinical trial.
So,
I think that's an important issue.
no,
We want to do this. I'm sure we've all got great questions.
yeah, so look, research funding, as you know, is changing dramatically in the next little while, particularly at NHMRC level. If you're looking more at, we now have the infrastructure. When we're talking about specific projects and specific ideas and funding for those, I think there are a number of opportunities and number of avenues that we can go along. Part of Vital is to help bring us all together to come up with the ideas and then perhaps direct us towards funding opportunities as well. Viive, I don't know if you've got any...
So the way we got this was by being collaborative, by having people from a lot of different areas come together and prepare this grant or be on this grant. And I think that's really how you need to move forward, to be multidisciplinary or to be attractive to funding bodies, be very much multidisciplinary. And highly translational and across a number of, what do they call those things, from the T0 to the T1 to the T2. Yes. So you need to, you need to have a lot of the pipeline there to be.
So, the question for Michael. I mean, is this something Tron might use?
I don't know. It's going to be up to individual clinicians and probably individual researchers. The scope of radiation oncology research around Australia is less so in the lab. It's more in the clinical settings. So it would be more the scientist giving a radiation oncologist on the tap to get something going from other centres. And I guess it's going to be part of the role of the Sydney Vital to advertise the facility so as then attracts researchers, know the facility's there, know what the costings are to do that. And I gather the Kolling's got such a good name in terms of small animal research and facility that the small animal researchers around Australia will be well aware.
Yeah, Dale, look, I think it's always a team effort and different people with different strengths come together. I think generally, I would imagine for this sort of research, translational research, I think looks good if the question comes from the clinic to identify a clinical problem. The clinicians can provide input onto some of the clinical questions and then work with the basic scientists to come up with an appropriate method and go from there. They certainly like the idea, in my experience, they often like the idea of a clinically based problem, a solution through clinical and translational research that will ultimately potentially pay out back into the clinic again. And definitely, I actually think we're going to be flooded with research opportunities with this piece of equipment. It's a huge piece. I think, whilst there's definitely been a lot of research in radiation oncology in recent years, this is a new era. This is a new era for medicine, and it's really exciting, and I think we should all get on board. And, if you ever, I'm sure there'll be, I would imagine, many budding RadOnc PhD students wanting to try and join the lab and do some research. And I don't know, if jobs were as hard to come by in RadOnc as MedOnc, they'll be lining up. And it's a great opportunity because, this is, a unique resource in Australia. There aren't many in the world. Because we've had clinicians that have had access to these drugs early, we've seen all these cases today that the rest of the world don't even know these problems exist. And yet we see them, we know them, and we've got an idea about how to change it. So, for example, Michael said, when you give SRS, it's worse. If you give, a short, course of maybe 5 treatments of fractionated radiotherapy, you get the same benefit from a control point of view, but maybe you minimise the tox. Let's test it in the mice. There's a lot of opportunity that's quite straightforward, at least on the clinical side, to throw the ideas around and then speak to the experts about how to try and model that.
And Alex, just to follow, the other thing I guess we've developed, Viive might talk to this, over the past 2 years has been a platform for looking at cell culture and exposure to radiation in collaboration with Michael's group. You know, we've been looking at, say, yttrium-90 beta radiation compared to external beam radiation. And we've got those assays going now. We've talked to Louise about moving from colorectal cancer to prostate cancer cells. And, you know, I think we're actually becoming a sort of a radio treatment testing kind of facility in some ways.
Absolutely, absolutely. And then, and again, a largely untapped avenue of medicine, really. Yeah. And a unique advantage we have here.
Yeah, and so the next step for all of that is to take it into animal models, which is why we have someone working in our lab on directly on that, and we will have other people from other centres doing exactly the same thing. But yeah, there's lots more opportunities there, and yes, you do need to get the funding, but it isn't a huge amount of funding initially, depending on what your question is.
And just nothing, just in the immunotherapy space, we have world leaders in animal research in Australia in immunotherapy, particularly up in Brisbane with Mark Smith, particularly, that we can collaborate with. We have, certainly our group have established collaborations. We can try and foster others. But I think the only thing that's limiting us, I think, is enthusiasm and sitting down and thinking about it. Because I think there's nothing like sitting in clinic and seeing these things happen to make you realise that there's a lot we can do. And I think it would be a shame if we don't use, you know, this wonderful piece of equipment to its potential. And I think I can imagine Viive would be saying that we need as much clinical buy-in as possible to try and make that all happen, and not just not just a single discipline. So we need a multidisciplinary approach to answering these questions.
Exactly, and yes, we've got some surgeons working with us now, which is great, and hopefully some rad-oncs and certainly med-oncs as well. So hopefully we can try and answer some of the questions and dilemmas that you're currently facing.
And finally, the one big question we have in immunotherapy is why do some tumours not respond? That's probably the biggest critical problem. And how do we make these sort of cold tumours hot again? And radiotherapy is one of the key things we're trying to answer there. And whilst there are people have jumped into clinical trials with conventional fractionated radiotherapy plus drug, we can do better than that. And I think this is a beautiful opportunity to do that. And as you said, you know, there are still ethics with mice work, I understand. We're still looking after them, but we will get the answers we need.
But we can sacrifice them at different. we can sacrifice them at different times so that you can then see what's happening in the tissue, what's happening in the tumour, what's happening in the brain, what's happening in the circulation, and we can do that at different time points as well as longitudinally. So yeah.
Any other questions before we wrap up? Any other?
Sorry, just a silly one. When I was doing PhD there, I always had problems anaesthetizing my pets, little pets. So what happens if the mouse just somewhere wakes up prematurely from anesthesia and just runs around the machine, inside the machine?
The door has to be shut anyway for the machine to work itself. So you can use a band to hold the animal in place, but you also have a camera directly onto the animal, so you can watch it in live sort of view to see whether its respiration is changing. With the anaesthetic machine, we can watch and see whether we're going to run out of anaesthesia or not. If the animal happens to wake up and move around the machine, obviously there's a shut-off that you can hit. The animal would have to be euthanized in that situation, though, because you haven't followed the correct procedure.
I've always asked, well, you know, we assume all the mice die. What if they get fixed, their cancer gets treated and they're cured? Do they get, like, put out to pasture or they get euthanized anyway? Yeah. Long-term follow-up with mice is about 20 days, isn't it, sometimes? We got 20 years. And then killed them. Yeah.
She's still alive.
They're at home, are they, with you? Okay, great.
What's her name? Beyonce? 26.
Number 26, lovely. Really attached to that one. Any questions, anyone? All right, well, look, I'd like to thank everyone for coming tonight.